Hello there, it's Mrs. Scoville, and I'm back today. We're going to talk about cell division. So we have 3 learning objectives for today's lesson. The first is to distinguish between mitosis and cytokinesis, identify the stages of mitosis by looking at pictures, and to name the hallmarks of each stage of mitosis in words, so to be able to describe what you see. We're going to do a quick recap here first. So a reminder, mitosis and cytokinesis are a part of cell division, which is just one part of a larger cell cycle. Mitosis and cytokinesis just make up the part where the cell goes from 1 to 2. All the other parts interphase lead up to getting ready for that cell division. We're going to watch a quick video here to recap that as well.
The orderly program of events in a cell's lifetime is known as the cell cycle. The eukaryotic cell cycle is divided into 2 phases. A brief mitotic phase, in which a cell divides to form 2 cells, and a longer period between divisions called interphase. Interphase can be divided into 3 distinct phases: G1, S, and G2. The G1, or first gap phase, is typically the longest phase in the cell cycle. Most cells will grow in size and manufacture a number of different proteins during G1. At the appropriate time, a molecular signal will cause the cell to enter the next phase of the cycle. During the S, or synthesis phase, the cell replicates the DNA in its nucleus in preparation for cell division. Precise control of the molecular machinery that replicates DNA ensures that only one copy of the cell's DNA is made during the S phase. After the cell has replicated its DNA, it enters the G2, or second gap phase. During G2, the cell may continue to grow and produces a number of molecules in preparation for the upcoming division. When the cell is ready, a molecular signal will cause the cell to enter the mitotic, or M phase of the cycle. During mitosis, the cell's DNA condenses into visible molecules called chromosomes. The chromosomes are carefully sorted between the 2 forming cells so that each cell receives one copy of every chromosome. At the end of mitosis, the cytoplasm is divided between the 2 daughter cells in a process called cytokinesis. Each of the daughter cells will enter the G1 phase of the cycle. Different types of cells exhibit variations of this basic cell cycle depending on their function in the organism. Some examples of different cell cycles can be found in the variations section. The progress from one phase of the cell cycle to the next is controlled by a number of proteins in the cell. To learn how these proteins direct this...
Okay, so that would be things like the cyclins that we saw in yesterday's lesson. So getting back to kind of the new part of the lesson here, so that was a recap. Reminder, just cell division is mitosis and cytokinesis, and mitosis is what we're going to focus on first here. That's where the DNA in the nucleus gets separated, and then we'll look at cytokinesis, where the cytoplasm gets divided. So again, mitosis is just simply and straightforwardly the part of cell division where genetic material gets divided evenly. Not worried about the cytoplasm, just the DNA. A couple of things have to happen before mitosis can occur, though, besides just going through G1S and G2. So in S, we know that each chromosome gets copied, and a couple of things here I just want to tell you terminology-wise. A chromosome, we know that that contains all of the DNA molecules. That's going to be something that's going to consist of these individual sister, what are called chromatids. So essentially, if we look at this over here, this chromosome is a duplicated chromatid. So each of these chromatids is a copy of the other. So these are identical. So again, they're identical. This is the copied state here. These are joined together. It's a structure called a centromere, and this is just the center of the chromosome. You can see they end up looking like an X, and during mitosis, we're going to see they're pretty condensed like this. So the reason that they're set up like this is when the cell separates, we'll see that these chromatids are going to get separated from each other, and this ensures that every new cell gets one of these chromatids, okay? So that keeps all of the cells the same, keeps all of your cells happy, they have all of the correct information. So just structurally, if you hear the term chromatid or sister chromatid, what we're talking about there is a duplicated individual like chromosome. So it's been duplicated and it's just a copy so that each cell will get one. Okay, so now we're going to dive into looking at mitosis, which is divided into 4 phases. So the 4 phases are prophase, metaphase, anaphase, and telophase. And if you remember PMAT, that will help you remember the order. So the first one here, prophase, the P in PMAT, there's quite a few things that happen in prophase. So first of all, it's the longest phase of mitosis, and I think you're going to see it's because so many things happen. So the first thing that happens here is the DNA is actually going to condense into those chromatids, like we saw in that previous picture. So this is where they start to take on that X shape. Prior to prophase, it's really going to look more kind of like spaghetti in the nucleus. This is when we start to see the defined X shape. So the next thing that's going to happen that's pretty important in prophase is you're going to have these areas called centrosomes, and they're kind of a region in the cell where you're going to have these other structures that are called centrioles form. And these centrioles are very important, especially in animal cells, because these are going to be the site at which an additional structure forms called the spindle. And the spindle, it looks like a little spider kind of coming off of the centrosome area, and again, we're going to have these centrioles. One thing to note though here is this is only for an animal cell. In plant cells, they have the centrosome area as well, but plant cells are actually able to make the spindle without having a centriole. So it's just kind of a little difference there. A couple of things to point out here in the picture though, besides the spindle and the centrosome area with the centrioles in it, is you can see here the nuclear envelope is breaking down as well. That's going to be critical to happen in prophase, otherwise we would never be able to separate the DNA later. It would be stuck. Alright, so next is metaphase, and this one's the easiest to identify. This is because all of the chromosomes with all those, again, paired sister chromatids, you can see them, are all lined up in the middle. So at the center of the cell, we call this the metaphase plate, or the center of the cell. So if you hear the term metaphase plate, it just means literally the middle of the cell. This is also going to be a very important stage because the microtubules that make up the spindle, so the spindle is just made of these structures called microtubules, which are also in the cytoskeleton, are going to connect to that centromere, so to that central structure of each of these paired sister chromatids. This is going to be essential as well to help hold them apart in our next step, which is anaphase. So here you can see the spindle is pulling apart these sister chromatids now into individual chromosomes. So this is going to set it up so that each of the cells will end up with one copy. And again, I've said here, they're going to continue to move until they've separated basically into 2 groups. So once they're in 2 groups, well, the cell needs to start to reassemble. So your last phase here is telophase or telophase, you can say it either way. And once they're separated, the chromosomes are going to gather at opposite ends, and they're not going to have that distinctive shape anymore. So here we saw they were kind of a V shape. Here they're going to start to look more like spaghetti again. You're also going to start to get new nuclear envelopes forming around those chromosomes, and so you get 2 nuclei. And finally the spindle will break down as well. So we saw just quite a large structure there. Now it's going to break down. So at this point, mitosis is done. So the final step that has to happen before the cells can be totally separate is cytokinesis. And with cytokinesis in an animal cell, what's going to happen is the cytoplasm is going to be pinched in half with something called a cleavage furrow. So it's just a structure that's going to form in a ring, and we'll see it in a little video here in a minute. And then that pinches them off into 2 separate cells. In a plant cell, well, we know that there's a more rigid cell wall. So what's going to happen there is actually just a new cell plate will form, and this essentially is a new cell wall. In both cases, though, I do want you to know that each daughter cell is going to have an identical set of chromosomes from the initial, what we could say, mother cell. So basically, each of these cells is identical to each other. This is very important to make sure that just that life proceeds normally, so that you have all the correct DNA in your cells. Okay, so now we're going to watch a couple of short videos to help recap this. Close that. So the first one here is an animal cell that's going to be undergoing mitosis.
This time time-lapse video follows an amphibian lung cell as it divides by mitosis. Our video begins late in prophase. The centrosomes, which organize the division process, are completing their migration to opposite poles of the cell. The paired sister chromatids that carry the cell's genetic material are clearly visible in the nucleus. Each centrosome sends out spindle microtubules in an attempt to capture a pair of free-floating sister chromatids. When microtubules from both
So it's a little bit hard to see here, but
they work together to move the pair to the cell's equator.
You can see that they're huge here.
Sometime after the last pair of
This is the DNA here in the sister chromatids.
A molecular signal causes the sister chromatids to become unpaired. This
The
is
spindles
beginning
here,
of
it's not really
anaphase,
stained.
in which an identical set of chromosomes moves to each pole of the cell. The chromosomes have reached the poles of the cell during telophase. At this time, the chromosomes appear less distinct as they decondense. New nuclear membranes can be seen forming around each group of chromosomes. A cleavage furrow can be seen developing at the cell's equator. Constriction of the cell's plasma membrane causes the cleavage furrow to deepen, eventually dividing the cell in two. This is known as cytokinesis.
Okay, so that was looking at an animal cell. Now we're going to look at it in a plant cell since there are a few little differences here. This is an animation, it's not going to look as real.
Mitosis is the process by which a eukaryotic cell divides into two cells. Each of these daughter cells contains an exact copy of the parent cell's chromosomes. Before mitosis begins, each of the cell's chromosomes is replicated or copied. The replicates remain attached to each other through early mitosis and are called sister chromatids. The beginning of mitosis is called prophase. In early prophase, the centrosomes move toward opposite poles of the cell, organizing the spindle microtubules between them.
So one thing I'm going to point out here is, again, in the plant cell, they still have the centrosomes, so those regions, but there are no centrioles. So there's not, that little structure is not in plant cells.
The chromatids become visible in the nucleus as they condense. In late prophase, the nuclear membrane breaks down and some of the spindle microtubules attach to the sister chromatids. The microtubules pull the chromatid pairs to the midline of the cell, or metaphase plate. The chromatids remain lined up between the poles of the cell during metaphase. Anaphase begins when the pairs of sister chromatids separate. The separated chromatids are now called chromosomes and move toward the poles of the cell. During telophase, the chromosomes arrive at the poles and new nuclear membranes form around them. Cytokinesis, or cytoplasmic division, begins when a number of small vesicles fuse together at the metaphase plate to form the cell plate. Eventually, the cell plate reaches across the cell and joins with the plasma membrane. This creates two separate cells, each with an identical set of chromosomes. The daughter cells are now in interphase between phases of mitosis.
Okay. So there's really only one other point to make here in terms of kind of why this is all important. So it's just a reminder that cell division is essential because this is going to replace dead and worn cells in our body, and remember, this is also how organisms are going to grow. We have to make new cells to grow. Also, if this goes wrong, people can get cancer. And also, if it goes wrong, your cells can die. So it's very important that cell division happens appropriately and correctly so that you can replace all of your cells. Also, interestingly enough, it also is going to be the mechanism that some organisms will use to reproduce. So they can just copy their cells. A lot of bacteria do this, and this is asexual reproduction. So that is the end of today's lesson. Make sure that you write your complete summary.